Good morning. Welcome to this session on replicability and reproducibility for machine learning applied to brain imaging. What I'm going to do is just go over some sort of motivating points for why we think this session is important, and then I'll introduce the multiple speakers that will be following. So in our goals for this session were first to create a bridge between brain mapping and machine learning statistical signal processing communities. This is one of the reasons we have Dr. Adali, who's the Vice President for Technical Directions for the Signal Processing Society. So she's going to comment on some of the algorithmic aspects as well as the data science initiative at IEEE. Objective 2 was to highlight particular ways machine learning approaches are have unique challenges in the context of replicability and reproducibility. And our third objective was to present some examples from experts in the field, ways in which these approaches have been used, and some of the challenges, some of the ways that we have to be careful. So in the context of machine learning and brain imaging in particular, obviously there's a huge emphasis right now on open science, on replicability, on reproducibility. However, most of the papers out there in this community have focused on standard pipeline-based analyses. So it's the garden of forking paths, for example. Also, all the other aspects of reproducibility, like being able to provide consistent code, being able to report your results completely. Machine learning approaches, I'm going to comment on one sort of small aspect of this, which is consideration for stochastic solutions that produce, say, slightly different parcellations, or algorithmic solutions that have different properties like uniqueness and identifiability. And finally, bias variance trade-offs. So you'll hear about all these things and more in the session. There will be 4 talks. I'll give a few sort of intro slides and some examples of different work that we've been doing. Tulay Adali will talk about algorithmic and machine learning perspectives in particular. Lisa Nickerson's going to delve into brain network estimation in more detail, and Gael Varoquaux is going to talk about going beyond reproducibility, looking at reusability and generalizability. So this is a very helpful figure from Christy Whitaker, looking at differences between reproducibility, replicability, robustness, etc. However, if we drill down into, say, the case where we have the same code and the same data, so that would be a reproducible scenario, and think a little bit about the context of algorithms that have that vary depending on random initial conditions, right? Or stability aspects or uniqueness aspects of algorithms, or pipeline properties, robustness to noise, etc. That adds another sort of layer into this in terms of how do we deal with those kinds of things. And I'll give some specific examples on that. Here's some FreeSurfer results. So FreeSurfer, we've been used for many, many years, but it is an optimization algorithm essentially, and it starts from a random initial seed. And if you simply vary that seed in the cortical parcellation and run FreeSurfer 10 times, this is an example of what you might find, where you can see on the x-axis I have different regions, and you can see the percent change goes up to close to 20% just across those 10 estimates. The absolute volume estimates vary by quite a bit as well. So obviously, if you're trying to develop biomarkers and you're trying to develop a parcellation that's predictive, and you have a new individual and you've run this algorithm on it, you can get a huge, I would argue, variation, even if you just run the software twice, everything else being the same. Okay, that's obviously not desirable. Here's an example from some work by Greg Kaar, where they looked at it a different way. They were basically adding stochastic noise elements to different parts of a pipeline to really look at how robust the pipeline was. And they found that there, not surprisingly, depending on where you introduce noise, the pipeline can vary in terms of its answer in comparison to the reference connectome quite substantially. And then thirdly, deep learning Here's a deep neural network, some work by Johannes Mehrer, and they're basically looking at a convolutional neural network, and you can see here that on the right side, if you just look from top to bottom, these are the different layers visualized in a two-dimensional plot, and you can see, just look at the bottom there, that things are not the same. And that's because it's, again, starting from different initializations, it gave different representations. In the end, it converged to different representations, both of which are potentially meaningful. So we can kind of get into that a little bit later. Here, I want to just highlight that big data, in the context of big data, it's challenging some of our existing rules of thumb. So, for example, you know, it's been thought that if we have more data per iteration, that's the best, you know, approach versus doing these sort of mini batches where we pull bits of data from everywhere and run it through a stochastic gradient descent approach. However, what we're finding is that if we use that latter approach, it converges faster, and it also scales to big data and gives you meaningful solutions. Second one, high dimensionality, meaning many parameters, will give you lots of local minima, right? So harder to optimize, more problems. What we're finding in actuality is that a lot of these are sort of so-called saddle points, and so if we correctly handle these, we can converge to quite good solutions, and we almost never get stuck in local minima. And then thirdly, bias variance trade-off. Obviously, we've been dealing with a lot of you know, parameter selection, you know, how many networks, a lot, a little, and we're trying to balance overfitting and underfitting and find kind of that ground that balances the two of those. However, you know, in this paper, they basically looked at this in the context of big data, and beyond a certain point, you actually see that the testing risk, meaning not generalizable risk, drops. It goes up to a certain point, and then if you add more data, it starts to drop again. So it's actually changing our view on even this sort of fundamental bias variance trade-off element. So, you know, we can kind of beat that with enough data. Okay, so now let me get to more specific brain imaging. I'll highlight a lot of ICA approaches in this talk. So just to kind of give you the history of ICA, you know, early on in the community, we focused on low model order ICA solutions. So between 20 and 40 networks, for example. And there are a number of approaches for estimating that as well. And that gives you these sort of large scale brain networks, fairly non-contiguous regions, similar time courses, et cetera. Importantly, ICA in most cases is a stochastic algorithm as well. So it is really important to deal with that in some way. One of the ways that we've dealt with that in the GIF software is we run it multiple times. We look at stability and we pick a solution that's sort of representative of you know, of the central point. The idea being that if someone else had the same data and ran the same algorithm, they would get a very similar or nearly identical answer, which in us, you know, which is a desirable outcome. So Tulay will talk more about this later. But since then, you know, things have pushed up to higher numbers of networks. So here's an example where there were about, I think this example here, we had 75, and there have been many solutions now going to 100, going to 200, et cetera. And so this is sort of blowing past what our bias variance estimate would give us, but these are really meaningful and informative decompositions because we are getting basically coherent subnodes within networks. For example, here in the visual domain, you see they're correlated with one another, but they're not identical, and they interact with different networks and different subnodes in different ways. Okay, and we're continuing to go up. So more recently, we've been looking at, you know, we're calling this ultra high order ICA, but essentially 1,000 components from the data set and trying to parcelate the brain. And we're seeing that evidence that there are meaningful and replicable solutions at that high model order. Here we've got 2 independent data sets, and we basically looked at the stability, and we found a little bit of a drop off at about 730 or so on 2 different independent analyses. if we compare with a null model in terms of stability, a random model in terms of stability, we find very much more stable solutions in the real data, suggesting that most of these components are reproducible. Here's just an example of just thresholded locations of the primary functional units on the cortex. And one of the benefits here is it's not to give us more variables, to just, weigh us down. It's actually really informative about the spatial temporal organization of the brain. Here you can see different parts of these. Once we get these parcellations, we can see how they contribute both spatially and temporally. So here we see multifunctionality. So regions that have higher values are contributing to more networks interacting over time. And we can also look at the dynamics of this, both spatially and temporally, and we see different reconfigurations. And so we see, for example, one of the components will jump between dominantly contributing to default mode, to an auditory network, et cetera. And this slide here is just to highlight the movie is not playing, but I guess that's okay. Essentially, we have three different aspects: temporal dynamic, where the coupling is changing but the nodes are fixed; spatial dynamic, where the coupling is fixed but the nodes are moving or shrinking or growing, etc., or splitting; and spatial temporal dynamic, where both of those things are occurring. And so it's really important to have these stable and data-driven parcellations in this context, because things can look dynamic in one way when they're not, if you don't have essentially a source that's coherent as the input to your node. And this is also true not only for resting fMRI, but it's also true for other modalities. This is just gray matter covariation. And so we've been looking a lot at this, and we've been also going up to higher model orders. So you see that the covariation of these gray matter regions actually groups into sets of regions that are remarkably consistent with what we see in resting fMRI data. They're not the same. They seem to be less specific, but it seems to be quite meaningful. And we've been going up to, in this case, we looked at about 15,000 individuals in our more recent results. So another way to deal with this, rather than just sort of testing stability, looking for consistency, is to use a sort of constrained approach. And so by that, I mean, you take some sort of a prior and you sort of regularize your solution. And so here's one example where we've been extracting component networks from large data sets. And so Human Connectome Project, genomic superstruct data. Independently, we found intersections between the two sets and move those forward as priors. And then we apply a modified ICA algorithm called spatially constrained ICA. And what that gains us is, A, it regularizes the solution; B, we get labels automatically for the components. We don't have to worry about labeling. And then C and D, it allows the networks to adapt. They can spatially vary among individuals. They can change, obviously, temporally. And we also get independent estimates so that we can use this for cross-validation and classification studies, et cetera. Just an example of what you see. We get highly consistent patterns of functional network connectivity and independent data sets. More interestingly, when we look at the more subtle control patient differences, we find also highly consistent differences. You can see, for example, these anti-correlations in this sort of cerebellum sensory-motor block are consistent in all three independent data sets. They're correlated, the most significant deviations are correlated up to around 0.9, 0.95. And we've applied this approach to make predictions that we think might be meaningful in a real-world clinical application. So, for example, predicting a response to medication. When you have individuals, young adults who might have bipolar disorder or major depression, you want to decide whether they should go on an antidepressant or a mood stabilizer. That's a challenging problem in some cases, and it can have real-world implications. People can get sick if they get on the wrong medication. And so using a pre-resting state scan, some work with Beth Osich, where she tracked individuals up to 2 or more years following to see which medication they ended up on that worked for them. And we found about a 95% accuracy to predict which medication they ended up on in the end that was the best. And this is regardless of whether they were on different medications at the time. And interestingly, also, we get these different networks that contribute most strongly to that prediction. Other examples, other ways you can deal with this are initialization. I don't have a lot because I'm running out of time here, but for example, the spatial prior of networks is one example of that. Another is just simply initializing if we're looking for dynamic connectivity states, for example, and we want to go to a new subject. And, do we want to just completely start from a random position, or should we initialize those states or those clusters to basically have the solution be comparable to an existing framework that we've developed or an existing database that we've developed? And I think there's a lot of extra things we can explore there. Just one slide on deep learning. I'm not going to unpack this. Actually, I could give multiple talks on this, but deep learning is a really rich modeling space. So we can look at these sort of spatial temporal aspects in really interesting and informative ways. And so in a sense, I think it's kind of missing the point to focus purely on accuracy in a deep learning model. However, even if you do so, you can see that, we see quite quite accurate results if we just look at some simple measures like separating age groups, right? Or separating males from females, for example. And if we compare that, across various numbers of samples to different algorithm solutions. So machine learning algorithm solutions like support vector machine, or kind of standard machine learning, even logistic regression or linear discriminant analysis, we consistently outperform in the case of structural MRI. And so lastly, I think competitions is another complementary way to really help us, because it provides a level of separation from the individuals who are doing the algorithms to those that are actually using them. We have done a few competitions over the years and many others as well. We started with this sort of Gael schizophrenia competitions, a few years ago. We have one ongoing right now, which is in collaboration with OHBM. It's the first UK Biobank competition, and it's also in conjunction with the IEEE Signal Processing Society. So there's $25,000 in prizes, and it's got about a few days left. It's going to be ending at the end of OHBM. But we have about 850 teams, over I think it's over 9,000 submitted solutions thus far. So it's been, quite remarkable how much activity we've seen. And so in summary, I hope I've kind of made you think a little bit about some unique aspects of machine learning and stochastic algorithms in general, and how do we deal with those in the context of brain imaging to assure we're getting solutions that are going to fit within our open science principles. We do try to release all of our software. All the approaches that I showed today are available on our website here and in various other locations. There are, I'm not really touching on it in these talks, but obviously how do you get these to run and scale these up and, get some software that enables you to do that easily and quickly and abstract it from the actual coding piece. There are a lot of solutions these days now. It's really exciting times. And, we're working on some as well, where we're trying to sort of encompass centralized repositories, decentralized repositories, provide dashboards, et cetera. So I think it's exciting to see where the field is headed, and I hope you feel that way as well. I want to thank the folks at the Trend Center who have contributed a lot to the work that I showed you today, as well as to the other speakers in this special session. I also need to mention that Dr. Stephen Strother is unable to present. He actually got stuck in New Zealand, and so without a computer, so he was unable to provide his talk. But Tulay, actually, I don't know if she's going to mention this or not. But he developed this framework called N pairs, which I would encourage you to look into. It's a really nice framework. And with that, I will stop. Thank you very much.
Another welcome from me to our mini symposium on replicability and reproducibility for machine learning with applications in brain mapping. As Vince mentioned, one of our objectives in putting together this mini session was to start a dialogue between the medical imaging community and the IEEE Signal Processing Society, which I currently represent as the VP for Technical Directions. We were planning gatherings for further brainstorming to see what future collaborative possibilities were there. In any case, I'm hoping to at least start the discussion, and I will definitely appreciate hearing from you. So please feel free to directly send me an email. I'm sure the virtual platform will also offer possibilities, but I really appreciate hearing directly from you as well. So what I'm going to talk about today is just bringing up a couple of main issues in matrix and tensor decompositions with a focus on ICA, which has proven quite fruitful for analysis of medical imaging data, in particular functional MRI, but for other types of medical imaging data as well. So to start, first, the question, why data-driven techniques? It's quite obvious. They allow us to make least amount of assumptions because we don't really know about if this is a multi-set scenario, what the interactions are. And with a data-driven solution, we can be fully multivariate, and we can also start with simpler models, assuming linear and multilinear relationships. And other priors, model-independent priors like smoothness, sparsity, independence, we can incorporate in addition. Now, blind source separation is a big umbrella of data-driven solutions, and inherently it emphasizes interpretability. Because in blind source separation, we start with a linear mixing assumption. So we have a generative model. So here in this one example, given the observations, we assume that we have two sets of matrices, factors, and in the spatial ICA model, here the rows of my source matrix, those latent variables, components, will correspond to spatial functional connectivity maps. And then the corresponding column here is going to summarize the temporal coherence of that given spatial map. Now, interpretability, we noted it's inherent in blind source separation, which means directly assigning a physical meaning to each row or column of my factors. Given the fact that I would like to interpret these components, obviously it's essential that I have to first make sure that this decomposition, these components, will be unique. Now, given that requirement, just a small reminder that matrix factorizations in general, they are not unique. So just if I start with this outer product type form, X equals AB transpose, I can always insert here a non-singular matrix C and its inverse, and I can define these two new matrices. So matrix decompositions are highly non-unique. Now, with ICA, I start with this linear mixing model, and the basic assumption is the underlying components, which I call sources within the ICA terminology, are statistically independent. Then, given the observations, those mixtures under this linear mixing assumption, I can uniquely identify these sources, subject to permutation and scaling ambiguity, of course, through a single demixing matrix, estimation of a single demixing matrix W. And this is true. I have these uniqueness guarantees for a large class of sources, including multiple Gaussians. So that's something I like to always underline, because it's typically noted that with ICA, we can only include a single Gaussian. That's not true. And I'll say a few words about that in a second as well. So I really have this uniqueness guarantees and for a large class of signals with ICA. But of course, I'm starting with the assumption that the underlying sources are statistically independent. Now, a very brief summary. ICA, indeed, it's not really a new technique by any means. The first approaches were introduced in 1980s. And then a decade later came two important algorithms, Infomax and FastICA, which were effective, worked for a good number of problems, and they really revived that whole ICA area, and a number of applications started very quickly, and the first ICA conference, and so on. Now, maximum likelihood estimation provides a nice broad umbrella for a good number of ICA algorithms. Why do I like that? Because then I can talk about large sample properties, optimality properties, and I can really talk about more a statistical big umbrella. Under this maximum likelihood umbrella, I can quantify the performance and can specifically say that the success of separation depends on how well I can estimate the underlying density of the sources, their distributions, and if I use multiple types of diversity, not only higher order statistics, which is the most common way to achieve ICA, but other statistical properties such as sample dependence, then I can achieve better performance. And this is also what enables identification of a wider class of sources, such as more than one Gaussian source. So given that Infomax and FastICA, indeed, they've been around for quite some time, and there are also a number of different flavors of those, but I would like to emphasize that even though still there is a tendency to go back to these old classical algorithms, now we have a good number of powerful ICA algorithms that incorporate multiple types of diversity and with flexible density models. So just to give an example on that, let's consider a very simple synthetic example with natural image sources. So these are my 4 sources. So here I'm showing that these have rich distributions. So they are not simple unimodal type density, but they have more rich overall distributions. So I'm just showing the simple histogram for these 4 sources. And this is showing the correlation, cross correlation across these 4. So here if we see the first and the fourth and the first and the third, so first and third, first and fourth, they also have some non-negligible correlation. So given this little bit challenging, not a whole lot, there are only 4 sources, scenario. So I have these 4 sources. I linearly mix them with a random mixing matrix. And here I have the performance of 4 different algorithms. So this is Infomax. This is FastICA using the flexible tangent hyperbolic nonlinearity. They do separate, and again, obviously quantification is easy, but because an image is worth 1,000 words, I'm just showing the estimated sources using these different algorithms, just to highlight the differences visually. So if I go to EBM, which includes a more flexible density model, I see that indeed the performance is clearly better. And on top of that entropy bound minimization density modeling scheme, I also include sample dependence. I see that the performance is much better, and actually they match the original sources quite well. Now, one other point here, these are all most consistent runs selected from 50 independent runs using random initializations. And I'll come to that in a second and will mention why it's important. Now, this is a very simple scenario, and for our favorite application, ICA of fMRI, the spatial ICA, comparisons are always difficult because for ICA, at least I start with the statistical independence assumption, but still the density models are different. So Infomax is using a very simple nonlinearity, which is a good match for super Gaussian sources, so peaky and heavy-tailed, which works quite well for most fMRI data. So EBM and ERBM are my more flexible density models. So here I have a sparse flavor of EBM and dictionary learning. So in this one example here, I'm just showing that, well, for default mode network, which is not necessarily super sparse, we have a better estimation. So here we are showing it with the circles, with, for example, the default mode network. And this is another EBM result showing that compared to others, we expect some modularity here. For example, here I have the different motor cognitive control default mode, but among those I see better modularity, which is another indirect way, because since all these algorithms end up with different decompositions, it's easier to look at them at a more global level for comparison. So more flexible algorithms indeed seem to provide a more useful decomposition overall. So after this very brief introduction to ICA, let's just come back to our definitions. So first, explainability versus interpretability. Now, explainability is the broader umbrella, which includes interpretability and also implies reliability of the solutions. So the final solutions that we obtain, they should be reliable, they should be auditable in a way. So there is a lot more we are asking when we want a solution to be explainable. So in that sense, this whole umbrella, reproducibility, replicability, robustness, and of course, ultimately generalizability, they are all implied under the big explainability umbrella, in addition to interpretability. Now, I put here 2016, because that's when, as we were discussing what we mean, and again, it's debatable. We might not be 100% happy with those, but this is what, again, now the National Academy also has accepted, especially for reproducibility and replicability. So this is a good working definition. So here, reproducibility, I have the same code, same algorithm, and same data. Replicable, if the code is the same, but my data is different. And for robustness, and again, each one of those, depending on where we are coming from, what our focus is, will have a slightly different flavor in how we interpret that. So for example, for me, robustness, the same algorithm. Let's say I picked one of those algorithms, Infomax. If I choose a different parameter, then I call it a different code, different algorithm in that sense. So for the first three algorithms I discussed, Infomax, fast ICA, for fast ICA, given that I already picked the nonlinearity, and EBM, I don't have any parameters except an order selection. So for most applications, we start with an overdetermined problem and then use order selection to decrease the order to project the data into a signal subspace. And for ERBM, in addition to order, I also have the memory length, but it doesn't really make a big difference. The default value works for most of those. So the good news is for ICA, everything is under maximum likelihood umbrella and information theoretic criteria. They provide a very nice disciplined way to principled way to pick the right order or to pick the order in a data-driven manner. And usually if we are within a standard deviation, the results don't change much. So it is quite easy and straightforward to select the order. So given that, I have fixed the order. So for these three and for this one, again, this doesn't make a big difference. But for those, I can definitely make sure that, okay, I don't have any user-defined parameters. So that's something I know for sure. So that's what I'm going to consider now. So I have the same code and the same data. So especially for these three, I have fixed the order and I don't have any other user-defined parameters. Now, initialization is the only other source of variability. And we do prefer random initializations. For example, I have the option of using identity or whitening type initialization each and every time. However, we and others have shown that if I start from different random initializations and I will have to pick the best one, so I'll come to that in a second, I am more likely to find a better solution because I have a non-convex optimization problem and using random initializations will help me get to a better local minimum. So in that sense, some variability is good. We prefer random initializations. I remember reviewing papers which said, well, we use JADE because each and every time I get the same answer. So getting the same answer each and every time might not be terribly desirable in most cases. So we can use multiple initializations, the more the better, depending on our computational resources. And most IC algorithms, especially these three, they are computationally super inexpensive. And then if I pick the right one, that's really going to provide a more consistent solution for me. So the question, how do I select one from these multiple initializations? So there are a number of solutions. This is something that we became aware of once we first started working on ICA of fMRI. So ICASO, that's the earliest method. So this is the fast ICA group they had introduced, which we integrated right at the very beginning, actually, within GIFT, Group ICA of fMRI Toolbox here. But then with multiple runs, we felt the need that we had to just pick a single run among those, not mix the components estimated from multiple runs. So this is the modified ICASO, just introducing a simple metric, which we introduced in 2011. And then we have the minimum spanning tree, which uses T-maps, and in a way that's quite a good match for the fMRI because we would like to really have those high T values. So it is very well justified, and it also provides a metric for interpreting the consistency of different components, so quite desirable. It's computationally costly, so no free lunch rule here. And more recently, a very simple idea, cross-ISI. So basically, if I have 100 different initializations, I pick the mixing matrix the estimated demixing matrix that's closest to all the other runs. And the ISI, this is intersymbol interference. That's how we evaluate the performance of IC algorithms using a single metric. And this is looking at the ISI. So this is the distance measure across multiple estimates of the demixing matrix. So taking all of those into account. And of course, all of those are included in GIFT. Now, one point to make. The most accurate run might not be actually the most reproducible. So this is quite obvious when we think from a generalizability point of view. However, it's a simple key point here. When we look at cross ISI, here what makes sense is to look at cross ISI as a function of ISI. So ISI is our global metric for evaluating the separation, the ICA performance. So the lower the better. 0 is perfect ICA decomposition, and 1, so it's a normalized quantity between 0 and 1, so lower the better. So if I look at cross ISI versus ISI, so here I have the Infomax results, and here ICA EBM. And this is a very simple unimodal, and in fact favoring Infomax a little bit. The sources are all from the generalized Gaussian density with a shape parameter that's matched to a super Gaussian. So it's a good match for Infomax. So it is, again, peaky with heavier tails, and 0.6 is the better match for the fixed Infomax nonlinearity. So I can clearly see that here the ISI value is lower than this case here when beta is 0.2. Now, if I look at EBM performance, 2 points. One, of course, here I have less variability. So in that sense, picking the most consistent run is a little bit less critical for Infomax, but more so for the more flexible algorithm, ICA EBM. Then, if I look here, Infomax, this is a good model match to the Infomax nonlinearity. So the ISI is quite low, lower than for the second case. But if I look at EBM here, so EBM does a very good job for both of those cases in terms of ISI. And in terms of cross ISI, here I'm showing all 3 most consistent run selection methods. So modified Picasso, MST, and the cross ISI methods. Obviously the cross ISI, because I'm plotting cross ISI here, is going to be here the lowest value. This is the magenta. But all 3 perform similarly when we have a good model match. So between the assumed nonlinearity and the actual source distribution. However, cross ISI is computationally very efficient compared to the other techniques, modified Picasso, Ricasso, and MST. And it's also preferable when there is poor model match. So it is a very simple and convenient way to select. And obviously looking at what the ISI is, that's something we can only do when we have generated data, when we are working with simulation examples. One question though, so if we look, this is 10 to the minus 4, 10 to the minus 3. So here this variability, even this variability, which is the biggest in this example, is not that much. So a question to ask is, are the final solutions really that different? Does it really matter? So I'm showing one example here from a multitask data. So here, this is a fusion example. It's a second level ICA analysis. So auditory oddball, Sternberg item recognition paradigm, and a sensory motor, 3 different tasks. So a data fusion example, one using the most consistent run and the other one the least. So we are just picking the 2 extremes, so that we call here the outlier. So it's not super easy here, but we see good agreement for the first set of components that are estimated, and then we start seeing here some differences. So if we just zoom in one of them, so the top one for AOD, this one component, if I select the one that's most consistent, that has the minimum cross ISI, I have statistical significance, and this is a default mode related component, so definitely relevant for the task. If I pick the one that's least consistent, has the high cross ISI, I do miss that component. And then I have this here, for example, another component which is quite robustly estimated, even though it's not significant. So indeed, picking the most consistent one really makes a difference. And because it's the one that is more interpretable, that also gives us additional confidence that indeed there is a good model match with ICA in these examples. Another example, so this one is a first level simple spatial ICA of fMRI analysis. So this is the distribution of the cross ISI values. Again, this is a normalized quantity between 0 and 1. So if I pick the lowest and here the highest, least consistent, and the most consistent run, which has the minimum cross ISI, and if I look at the power ratios of the estimated components, again, I see that these are higher, saying that, okay, I'm estimating more bold activated, bold related components if I select the run with the minimum cross ISI. So a few concluding remarks. So first, I noted if I'm going to interpret the components, which is natural with blind source separation under that umbrella, I would like to make sure that my decomposition is unique. I have some uniqueness guarantees. And with ICA, we have it under very general conditions. Of course, I start with the assumption of independence, but after that point, especially if I use multiple types of diversity, statistical property, I can identify a very broad class of signals. Now with other methods, dictionary learning, which also has found good use for fMRI analysis, for example, in addition to many other applications, or I have non-negative matrix decomposition. For these, the uniqueness guarantees are more limited. For example, with dictionary learning, I have to specify the level of sparsity. They are not as general as in the case of ICA. With tensor decompositions, I do have with the second order decomposition, for example, with the CP decomposition, uniqueness guarantees, but of course, provided that I know what the rank is. So in that sense, I would like to emphasize that indeed, starting with uniqueness guarantees is important. However, even with that, we noted that we have to be careful. So one other point to underline is that the most accurate run is not necessarily the most reproducible one. So this is a point that I end up discussing quite a bit with colleagues who work on more algebraically motivated methods. So for me, dictionary learning, tensor decompositions, even though obviously we can make the statistical connection through the Gaussianity assumption, they are more algebraically oriented. But picking the most reproducible, most consistently estimated one is the one that's going to give me generalization. I won't say guarantees, but will provide more confidence for better generalizability. So something we have been doing more recently now, looking at subsamplings. Always we used to say more the better, but now we are working, well, what if we actually look at smaller N studies, is the decomposition stable? So in that sense, more might not always be better because I would like to better generalize. So in that sense, it's also important to determine the sample size small enough that will guarantee us some generalizable results. So it's a new effect size, if you will. So this is for replicability, an important consideration. Then, so within deep nets, machine learning, those hyperparameters, user-defined parameters. So with ICA, apart from order selection, we don't have any of those, but still we noted that we need to be careful because initialization makes a difference. We would like to make sure that we are going to find a more repeatable run. And obviously this is a more difficult case for other algorithms. For example, with dictionary learning, NMF, I am actually estimating both matrices, not just one, the mixing matrix as in the case of ICA. So order selection is a more difficult problem. For tensor decompositions, rank estimation is difficult. And obviously with dictionary learning and other general matrix decompositions, I also have to work on those regularization constants and so on and so forth. So for ICA, things are much easier, but still we have to be careful. And this is one of the notes in all the reports about reproducibility. So we really need to educate. We really need to clearly transmit the user all the information. And one of those, it seems like a small point, but it can make a big difference. We really have to run the algorithm multiple times and make sure that we select the one that's most consistent among those. So I'm going to thank my group. So this is a recent photo op, and again we have our codes, publications on our webpage, and I'll be happy to hear from you, any comments, questions, and future collaboration possibilities.
Hi, I'd like to talk about better machine learning in brain imaging, and I'd like to move the focus from reproducibility to reusability and generalizability. First, I'll talk about the problem of non-tested generalizations. Then I'll talk about usable machine learning for brain imaging, and then I'll mention my dreams about better generalization. So what's the problem of non-tested generalizations? Any scientific claim must generalize. For instance, biomarkers of pathologies should carry over to new acquisition sites. Models of mental processes must apply to new stimuli, and cognitive theories should generalize across psychological manipulations. So claims of generalization should be explicitly tested. For instance, generalizing from one paradigm to another. For evidence of generalization, machine learning practices are required. We need to test quantitative model predictions on new data. Typically, you take the full data, you divide it in a trained set, you fit a model, and then you explicitly test the predictions of the model on a test set. This is called cross-validation, and the way to do it right is to repeatedly split out 10 to 20% of the data as test and not usually one out cross-validation, to split the data in a way that tests a real generalization. For instance, to do subject diagnostic, no image of the same subject should be in the train and in the test set. Importantly, you should not tweak hyperparameters or make choices to improve the score. This requires nested cross-validation or train-test validation split if you need to make model choices. And you should have at least 300 observations to measure correctly the test's accuracy. In general, the problem is that cross-validation is not reliable with small samples. If I have 300 samples, this is the distribution of test error that I'm observing due to sampling noise. Sampling noise is basically the changes that I observe if I do random choices of subjects. Now, if I have more observations, the sampling noise goes down, but you can see that at 300 observations, it's still quite wide, plus and minus 5%. Now, the fact that we're making modeling choices which explore analytical variability make things worse. If I do trivial analytical variations on a permuted data, for instance, changing smoothing, the choice of a decoder, or feature selection, I'm observing the huge variability of cross-validation score that I'm showing on the figure here. So on the data that is actually permuted, for which there is no possible prediction, I can observe to as much as 71% chances, 71% prediction score. So with a small number of observations, some analytical choices will give seemingly good predictions by chance. In practice, this lack of control undermines published work. In the literature, reported effect sizes decrease with sample sizes, as you can see on this figure, which was done via literature survey. These problems have been observed by other researchers studying more specific problems, such as performance of machine learning in neuroimaging studies of depression. So, machine learning brings the promise of reliable evidence for generalization, but it needs to be used well. And for this, we need good tools. To facilitate good machine learning practices in neuroimaging, we need first to democratize machine learning. Papers are not good enough for this, and software silos create friction. We also need to regain trust in methods, the thousands of methods that are published yearly, and people actually use what their friend used. So we need more than code supporting a publication. We need shared, open, and reusable software tools. I dream of an open, modern computing environment with no licensing or installation problems that can be used in the cloud, for instance, to support neuroimaging tools such as NeuroVolt, NeuroSynth, or NeuroQuery. The growing Python neuroimaging ecosystem is providing this tool. Python is a popular language, as the Google search show. It's also more and more popular for medical research, biomedical research, as the PubMed counts show. Historically, neuroimaging has been driven by MATLAB and exome tools like SPM. However, as you can see on this graph, Python is increasingly used due to exome tools such as NiBabel, NiLearn, DiPy, or MNePython. NiLearn provides statistical learning for neuroimaging in Python. It does all the steps of a pipeline after preprocessing, extracting signals from the brain images, visualizing brains, surfaces, connectomes. Recently, we just added standard GLM-based task modeling. We have a full functional connectivity pipeline, and we can do decoding, that is prediction of behavior and phenotype from brain images. We focus on being open source, easy, lightweight, to facilitate experimenting and teaching. It's also paired with scikit-learn, which is the reference machine learning toolbox. All the documentation of NiLearn comes with neuroscience examples that are runnable online. For this, we need open data. So we focused on finding good open neuroimaging data. And we need fast computation. So NiLearn tends to find good algorithms and good trade-offs. This forces us to distill the literature just as if we were doing a review, except what we're providing is code that support this review. So with this, I dream of better generalization in neuroimaging. We have a bunch of challenges. We often have unfalsifiable overgeneralizations. We have sample sizes that are dangerously small. We have lack of consolidation across neuroimaging findings. And we have an increasing analytical variability. We can tackle unfalsifiable overgeneralizations by turning them in testable prediction. For this, we need strong generalization, more than cross-validating a study. For instance, instead of claiming area A was recruited in task B by mental process C, we should show that area A predicts mental process C across many tasks. This is doable. It requires machine learning across tasks and studies. Our sample sizes are dangerously small. To tackle this problem, we should reinforce a given study with prior data. In machine learning, this can be done with transfer learning. And we've shown that we can leverage implicit similarities to previously studied conditions to improve decoding in a given condition. The important aspect is those similarities are implicit. We don't have to specify them. There's a lack of consolidation across neuroscience findings. We should make quantitative predictions from literature. NeuroQuery does this. You give it a verbal description of a neuroscience question, it predicts from the literature the brain locations of the corresponding findings. It's also available as a web service, which you can use online. We're facing increasing variability as we publish more and more methods. We should benchmark those methodological choices across many data sets, and we should come up with recommendations. My group is providing certain recommendations for decoding methods, for functional connectivity methods, for brain parcellations, which we have all established on many open data to be challengeable. We need more of these studies. So I dream of better generalizations and better machine learning in brain imaging. Generalization must be tested with solid cross-validation and not just asserted. Open, usable machine learning tools facilitate and enable generalizable findings. And we need new, better methods and better studies that generalize across prediction. Thank you.
So my talk is going to focus on reliable and reproducible brain network estimation. There are common research questions regarding brain networks that focus on network discovery. So identifying this repertoire of brain networks that may be active during rest and during different tasks, and then assessing intersubject variability in connectivity. For example, assessing group differences in brain network connectivity between patients and controls. More recently, though, there's a growing interest in single subject level network estimation. These maps reflect individual variability in topology and function and can play an important role in clinical settings for personalized diagnosis, prognosis, and treatment. They can also play a role in biomarker development and other applications. So for this talk, we'll be focusing on these large-scale networks and brain network estimation of this at the group level and at the single subject level. There are two popular frameworks for brain network estimation. The first is regression-based framework, which is hypothesis-driven, flexible framework in which a seed region of interest is defined a priori that you want to study the connectivity of with the rest of the brain. The strengths are that it's straightforward to implement and it's straightforward to interpret the results. You get a subject level seed connectivity map that shows the different voxels in the brain that may be connected with your seed region of interest. But can we use these maps to identify interpretable, reliable, and reproducible brain networks at the individual or group level? Some issues to consider regarding interpretability are that the spatial covariance between voxels is ignored in this estimation. The seed connectivity map is an aggregate connectivity map that can reflect multiple networks that share connectivity with the seed ROI, and it's not possible to disentangle those connectivity maps into the individual networks connected with each seed. This also only examines the connectivity of a single region of interest, so it doesn't inform connectivity of other active networks that are not connected with the seed. And there's potential for seed selection bias in which very slight changes in the location of a seed, say within posterior cingulate, can give you very different connectivity maps. There are methodological issues that impact reliability and reproducibility too. So functionally inaccurate ROIs and acquisition parameters like the length of the scan are critical, with longer scans improving sensitivity to detect brain connections and increasing reliability. Pre-processing choices have been shown in numerous publications to strongly affect these connectivity outcomes as well. A 2019 meta-analysis and systematic review of nearly 70 functional connectivity papers using these regression techniques showed that individual edges show marked instability with poor ICC. Within network connectivity was most reliable, with frontoparietal and DMN being the most reliable networks and subcortical networks being the least reliable. Eyes open rest, smaller interscan intervals, using full correlations with shrinkage and longer scan times improve reliability. And these results from this 2017 paper really show that scan duration is critical. Even to achieve a fair reliability in these cortical regions, you need more than 10 minutes worth of fMRI data for each subject, and you need more than 35 minutes to achieve fair reliability in subcortical brain regions. So this is a critical parameter. Successful denoising reduces reliability by reducing systematic unwanted effects such as motion. So while reliability is reduced by denoising, validity is greatly increased in the resulting connectivity maps. So these authors make the point that care should be taken in promoting reliability at the expense of validity. Spatial ICA of fMRI data is the other popular technique for brain network estimation. This can be done at the single subject level shown in this figure, or it can be done at the group level by temporally concatenating the multi-subject data prior to the ICA. ICA effectively separates signals from noise and disentangles brain networks. However, model order is critical as it determines the granularity of the functional organization that's being studied. In other words, identifying 20 to 50 components from a group ICA will reveal components that illustrate these large-scale brain networks. Whereas if you increase the model order, each of these components may just reveal small subnetworks or individual brain regions. Challenges of ICA for network estimation beyond determining the appropriate model order is that ICA is a stochastic iterative algorithm. So slightly different solutions are obtained each run of ICA, but you can use Akaso to assess the stability of the decomposition. It may be that the stochastic nature is more important for single subject ICAs versus group ICA. For single subject ICA, component matching is also challenging. So identifying a default mode network, for example, from single subject ICAs across different subjects can be a very difficult problem. So in that sense, most functional connectivity studies implement group ICA to improve the estimation by virtue of using more data and to enforce correspondence across subjects. So you get these nice group level network maps, and then you can use back reconstruction for subject level network estimation. Now there's a convergence of a large body of research that shows that large-scale brain networks from group ICA have been reliably and reproducibly identified across numerous task and resting state fMRI studies. So even the very earliest applications of fMRI to task, of ICA to task fMRI data at low model order revealed a collection of visual networks that were modulated by the task and other networks that were not modulated by the task that we now think of as resting state networks. A large repertoire of resting state networks have been identified very consistently in lots of different papers on functional connectivity. And these resting state networks encompass many different functional domains, including visual, auditory, motor, and neurocognitive domains. And again, this is at lower model orders. In 2009, Smith showed that these same resting state networks observed at lower model order also correspond to the repertoire of task networks that we use to perform everyday tasks. This study, this 2009 study, was fully replicated in 2018 using different data and analysis methods. And if you look at the resting state networks on the left-hand sides of each of these panels and the task networks on the right-hand side, you see strong spatial similarities between the resting and the task networks. And we see this replication across these two different studies. So at low model orders, we can really obtain these highly reliable and reproducible large-scale brain networks. And so that's working in this regime of ICA decomposition, where we see default mode networks and visual networks. As we increase the model order of the group ICA, say over 100 to 200, the resulting component maps reflect individual brain regions or very small subnetworks. But the reliability and reproducibility of higher order model ICA has not been well established. So for group level network estimation, seed connectivity shows fair reliability for frontoparietal and default mode networks and poor reliability for the other networks, especially subcortical at the individual and group level. Group ICA produces highly reproducible and reliable network maps. But what about subject level network estimation from single subject ICA and from group ICA, in which we use a back reconstruction technique like dual regression or PCA based back reconstruction to identify these subject level network maps corresponding to our group networks? Group ICA with dual regression has been shown, studied, reported in a couple of different studies. Large-scale brain networks have moderate to high reproducibility and replicability, and moderate to high short and long-term test-retest reliability. The dual regression subject-level maps have modest reliability and reproducibility of subject-level networks that was shown in both of these studies. Single subject ICA showed limitations for template matching procedures to identify the subject level networks. And this plot just shows that the inter-session ICC and intra-session ICC were highest for the default and control networks. One study compared seed-based connectivity with group ICA with dual regression, which is kind of an apples to oranges comparison. But they did find that the percentage of reliable voxels in 7 brain networks was much higher for group ICA with dual regression than for seed-based connectivity using a posterior cingulate seed. A study of group ICA with PCA-based back reconstruction found high within subject inter-session reproducibility of spatial maps across 158 sessions of scanning data from a single subject. But the reliability of the amplitude of the time courses in between network connectivity measures was low, except for the left and right executive networks. And the subject level maps, network maps showed high spatial similarity to the group mean. So one thing I would like to point out is that all these studies assessing subject level network maps use different metrics, and so they may not be, results may not be directly comparable across the different back reconstruction methods. So in conclusion, group level large scale networks are highly reproducible and replicable from lower model order group ICA. Using seed-based connectivity methods only achieves fair reproducibility and reliability of frontoparietal and default mode networks, and it's poor for other networks. At the subject level, brain network estimation using seed-based connectivity results in poor reliability of edges and modest reproducibility and reliability with group ICA with back reconstruction. So maybe we can look to other methods for subject level network estimation that have great promise to improve reliability for these kinds of studies. So constrained subject level ICA techniques, other machine learning methods, and even incorporating stability measures into the estimation for subject level ICA and for back reconstruction may all lead to improvements in subject level network estimation. And the last thought that I would like to leave you with is that reliability, validity, and utility are all important. So denoising reduces reliability, but it greatly enhances the validity of the connectivity results. Although the reliability of subject level maps ranges from poor to good for back reconstruction, dual regression is based on a generative model, and both back reconstruction methods are sensitive to meaningful intersubject differences. And this was studied in depth in this Allen 2012 paper. And then the last comment is that there is new emerging evidence that prediction of behavior using connectivity features is more reliable than the individual features alone. So this is very encouraging for studies at the single subject level. So thank you.